last lecture focused on the cell membrane, its structure and its function. And if you remember, we mentioned that there were three different functions, right? And we talked about 1 and 3. We talked about how it serves as the boundary, and we talked about cell-to-cell communication. So today we're focusing entirely on transport. So how things get across the cell membrane, aka how things get into and out of a cell. Okay, so before we discuss this, we need to kind of discuss a more general concept, which is called a concentration gradient. And so what that means is that if you think of a cell and if you think of what's outside the cell and inside the cell, we've got different stuff there. Okay, that stuff is called solute. It's just stuff that's dissolved. So that stuff could be sugar, or that stuff could be salt, or it could be oxygen, or whatever. Okay, so we've got different concentrations of, let's say, oxygen inside the cell and outside the cell. And if there's a difference in those concentrations, that's called a concentration gradient. And generally, molecules are going to move down or with the gradient from an area of high concentration to an area of low concentration until it equals out, okay? So right here in this picture, there's more green dots of solute, whatever this stuff could be. We could call it sugar. Okay, there's more sugar outside than inside. And so the sugar, this has to do with like intermolecular forces and fancy pants chemistry, physics stuff that we're not going to get into. But basically, naturally, these things are going to want to equalize. And so the sugar is going to want to move from outside the cell to inside the cell until we get like even amounts. Okay, but it might not be able to do that. Okay, something might keep it from doing that. So, for example, the size of the molecule is going to come into play. So, you would imagine here that these phospholipids are packed pretty closely together. There's some little gaps, but they're pretty close. So, if you have a large molecule, it's not going to be able to fit in between the individual phospholipids. Also, if something is polar, it's not going to be able to go through because guess what? Even if it's teeny tiny and polar, most likely if it fits through here, all of a sudden now it's in this non-polar region and it's going to get kicked back out again. Okay? Or if something has an electrical charge, again, it's not going to make it through the interior. It's going to get kicked back out. Okay? So that might prevent things from just freely passing through the membrane. Also, some cells or things are set up to have a gradient. So if, for example, your nerve cells work, the whole reason why they send messages is because there's a different concentration inside and outside. Mitochondria need this, chloroplast needs this. So sometimes that difference is going to be beneficial. Okay, so concentration gradients differences are not always bad. Okay, sometimes they have a really good purpose. So when we're talking about transport into and out of a cell, there's two main types, passive transport and active transport. And we're going to go through both of those individually. Passive transport, the reason why it's passive is like basically there's no energy required by the cell. And cell energy is called ATP. Okay, so no ATP is required. The cell doesn't have to do anything. And so this is the substance diffusing from an area of high to low. And they're just going to move naturally on their own until equilibrium is reached. Equilibrium means equal amounts. And then like for every two that come in, two that'll go out. A little back and forth will happen. Okay. And this is how a lot of materials come into your cells and also how we get rid of wastes in this way. Okay. So here's an example, right here, CO2, that would be like a waste product. Right here, there's a high area. It's going to just diffuse across the cell membrane to an area of low because CO2 and other things that are small or uncharged and nonpolar can just move right through the membrane. And CO2 is an example of that. Oxygen is another example, something that can diffuse right through the membrane without any other action or energy. See, high area, low area, here it starts diffusing, and then at the end, we end up with equal amounts. Okay. Now, this is a more specific type of passive transport. This is called facilitated diffusion, and if you remember from our membrane structure lecture, there are these proteins embedded in the cell membrane, and some of them are like open tubes, like this one right here, channel protein. And those open tubes are basically like almost like a donut or like a tunnel through the cell membrane. And so remember over here when I talked about, hey, what if the thing's too big? It can't get through. Well, it can use a little protein door like right here. And so this provides a little doorway through the membrane. Okay, so what facilitated diffusion is just when you need to use a protein to move something across. This is still passive, so no energy has been used. Okay. And you use this for large molecules or for something that's charged or polar. But again, we're still going from high to low. We're not using any energy. And this could be just an open doorway, like a tunnel, which is what this one is. This one's like a tunnel. And this one is called a carrier protein. And this one's going to slightly change shape. So like when this guy comes in and bumps this thing, now it's going to bloop, close at the front and open on the bottom. And although that might seem like it uses energy, it doesn't use any energy from the cell. And so this is also another way that a protein can be used to get something across the cell. This is almost like a revolving door in like a shopping mall or something, that's kind of like that. Whereas this is like an open door. Both of these are passive. Again, high to low, not using any energy. Kind of a subset of that is osmosis. So when we're talking about water traveling, we have a different word for it. And so osmosis is specifically the diffusion of water. So we're still talking about something going from high to low. Okay. Now, water can move through the phospholipid bilayer. It's small, but it's also polar. And so a lot of it gets kicked back. Again, like we talked about, it's going to get to this middle and get kicked back. But some water molecules will make it through. Not very many, though, because of its polarity. And so actually water ends up traveling through by facilitated diffusion with a protein, a channel protein that's like an open door, like a tunnel. But of course, because we love special names for things, when we're talking about water specifically, this process, osmosis, okay, they use these proteins called aquaporins, like aquapore, right? Okay, so that's the special protein that lets water through. But again, it's going to go from high concentration to low concentration and not going to use energy still because it's passive. So next we're going to talk about active transport. Active transport is using energy. Why? Because we're going from low to high. So if there's already a lot of something in that area and you're going to put more of it in that area. So this is going to set up that gradient that we talked about. This is going to set up a difference between inside and outside. And this is going to require a protein, okay? And these proteins are going to move the thing across the membrane, and the proteins are going to need ATP to work, okay? Which makes it different from here. This protein did not need ATP to work, and this protein did not need ATP to work. But this one does. So if you take a look, notice outside there's a lot of yellow dots. It doesn't matter what this thing is. It's something that there's a lot of, and there's very little here. But let's say we need even more of it up in this blue area. So the yellow goes in, and then ATP binds, and then the door releases it to the other side. So this is going to pump more of the yellow stuff, even though there's already a high concentration here, okay? So in order to change this shape, a shape change, the fancy pants way to say that is conformational change. In order to get that shape change, ATP has to bind, okay? And actual real life example of this are sodium potassium pumps and also proton pumps. But we're going to look at sodium potassium pumps. And so what a sodium potassium pump does is sodium is going to go one way, potassium is going to go the other. So basically what happens is we get 3 sodiums going in, okay, and an ATP binds, and then ATP splits, and that's going to release our 3 sodiums, and that's going to bring our 2 potassiums back in. So 3 sodiums going out, 2 potassiums are coming in with this pump. So we're going to be like increasing sodiums up here and increasing potassiums down here. And as it turns out, this is how like your heart beats. And this is how messages are sent on your neurons. So like if you put your hand on a stove, this is how the message gets sent, and then it comes back to you like, take your hand off the stove. All of this is from sodium and potassium pumps, okay? Your kidneys use this as well. So let's think about the charge here. If we have 3 positive sodiums, let's say the blue is going out, and 2 positive potassiums coming in, what is this going to have on the overall charge of a cell? 3 pluses go out, 2 pluses come in. That means the outside is going to be more positive than the inside of the cell. So we're going to have a different charge on the inside and outside. But again, that's how like neurons and hearts work. So that's good for them. So that's active transport. Whenever we are using some kind of protein and ATP to move from low to high. Another type of active transport is this bulk transport. Let's say we want to get super duper huge things through, and the things are so big that they won't fit through a protein. The protein is not big enough. It would be like trying to fit a semi truck through a regular door. Not going to work. Well, what do? Your cell does what's called endo or exocytosis. Okay. So with endocytosis, this is a big molecule that needs to come in. And so it meets up with the cell membrane, and the cell membrane actually pinches around it. And this little like part like bubbles off and becomes a vesicle and travels through the cell to where it needs to go. Exocytosis is the opposite of that. You're going to have some type of vesicle inside traveling. It'll fuse with the cell membrane and become one with the cell membrane again, and then release that to the outside of the cell. Okay. Again, this is active because it isn't shown in this process, but to like fuse these vesicles and pinch them off requires some cell energy there. All right. So here's a nice little summary. Okay. We've got diffusing from high to low. If it goes straight through, this is passive. If it goes through, but it needs a protein helper, but we're still going from high to low, this would be also passive, but specifically facilitated diffusion. Okay. Here's active going from low to high, and ATP is being used here. Again, another really good summary showing that simple diffusion is nonpolar, hydrophobic things, small things. They go from high to low. Facilitated is going to help those bigger things, maybe hydrophilic things, things that are polar, still high to low, but we're going to use a protein. And then active here is we're going low to high, definitely using a protein that's going to pump some things, and it's going to require your cell to use up ATP. Here are our things that you should know about and be able to answer by the end of this lecture. And here's where we are in the book.